All right. Okay. Now I think we can start. All right. So good morning, everyone, and welcome to Neuroccino. It's 9:30 in the morning of the last Monday of June. I'm a bit sad today because it's our last Neuroccino before the summer break. But yeah, let's recharge our energy for the next season. Okay, so without wasting any more time, I present our speakers. So today we have Marcella, postdoc from Bordeaux, and she has an interesting paper on chimpanzee and humans. So now, 1 second. Okay. Now you should be able also to share the screen.
Okay. So yeah, hello everyone. Can you see the paper?
Oh, yes.
Yes. Okay, good.
And if you can also share the link. Perfect.
Yeah, I can. It's not open access, so I don't know if you will be able to read it online, but of course I will share the link. Let me just find it. Wait, please. Copy. Okay, where is the chat? Sorry, I'm just trying to go to the chat. I lost the Zoom.
Yeah, no problem.
Chat. Okay, here. Okay, so there is the link. It's not open access, so but then if you are interested, I can just send you the paper. So yeah, as Sabrina said, today I'm presenting this paper that was recently published in PNAS. So this is entitled Human and Chimpanzee Share and Divergent Neurobiological Systems for General and Specific Cognitive Brain Functions. So in this paper, the author compare data from humans and chimpanzee with two aims. So in the first one, they explore the networks of brain connectivity that were associated with non-social cognitive skills, such as problem solving and relational reasoning, that are overlapped between humans and chimpanzees. And then in a second analysis or second part, they explore whether there are some brain circuits involved in a specific brain function and cognitive domains that are relatively well developed in each of these two species. So for the method, I will go directly to figure 1, in which they have this scheme of the methods. So for humans, they took some data from the Human Connectome project, in which they analyze 480 data from taking into account the T1 and diffusion weighted imaging data, and also different cognitive tests that I can show you later, in which they calculated a composite cognitive score. And in chimpanzees, they took the data from 45 chimpanzees, and also they took some data from cognitive tasks. I can just go I'm going to the method just to show you some of the different cognitive functions that were explored. So here we can see in the my right, some of the cognitive functions that they explore in the different tests in the humans. So in general, they explore executive functioning, processing speed, episodic memory, and working memory. And they calculated these composite cognitive scores. So at the end, they only had one score per subject. And then for the chimpanzee, they explore this primate cognition test battery that explore what they call physical cognitive capacity. And these are supposed to measure similar cognitive functions in both humans and the chimpanzees. And also they explore some social cognitive capacities, but then they didn't explore deeply these cognitive functions, and I will tell you why. And they also calculate this composite cognitive score in the chimpanzees, which involve all the different tests available in this primate cognition test battery. So also what they did, they took an atlas to define different regions of interest. So I'm going back to the figure 1. So what they did, they take this atlas and define the region of interest, and they use the weight of the different tracks to define this link connecting the different regions of interest. So they used an approach that is called connectome-wise association, in which they explore the correlation between these different weight of the tracks and the composite cognitive score. And then what they did, they kept all the coefficient of correlations that were above a specific threshold. So they defined the threshold as the 29% of the links that have the strongest correlation coefficient of correlation. So they did this in half of the data set, so they call it the discovery set. And then they multiplied this coefficient of correlation in the data of a new data set, so the testing set. And what they did by this step was to generate a predicted cognitive score. So this is what they call polyconnectomic score, that is supposed to be the predicted cognitive scores of this testing group. And then in order to validate if this prediction is correct or not, they run a correlation between the predicted and the real measures of this cognitive score. And if the prediction is okay, it's correct, we will see a positive correlation between both. And then finally, to test if this network of brain connectivity was also able to predict the scores of cognition in the chimpanzees, they applied, same as for the human testing group, they multiplied the coefficient of correlation obtained from the first group with the brain networks of the chimpanzees. So they obtain this prediction of the cognitive score, and they observe if there is a correct prediction or not. So this from A to C is the method they use for the first part of this paper, that was to explore if there are some common networks between humans and chimpanzees to predict different cognitive scores or different cognitive abilities. And then in a second part, so in D, the second part of the method, they took some brain maps for language and working memory, and also for different terms related to cognitive functions in the database of Neuroccino. in which they calculated the network strength of the networks in humans and chimpanzees. And what they did, they compare if these networks were more strongly connected in the brain of the humans or chimpanzees. So for the first exploration, so what we can see here, the results in the figure 2. So we can see in the left, they have the top most strongly associated connections that that are the coefficient of correlation when trying to predict from brain the cognitive scores. And we can see in the right that from the first group of the human data, they were able to predict the cognitive scores in the testing group. So we can see that these are the coefficient of correlation between the predicted score of the cognitive scores in the testing group. And here in the x-axis, we can see the real cognitive score. So we can see that there is this positive correlation that is significant. So they were able to predict the scores in this group. And then in D, we can see that it was also the case for the data in the chimpanzees. So what they conclude from these first results is that based on the one network that is important in humans to predict cognitive scores or cognitive abilities is also important or is also significant to predict cognition in the chimpanzees. So they, from these results, they also can conclude that these are similar cognitive anatomical connectivities that are related to cognition in both species. And then for the second analysis, we can see here in the figure 3, what they did, again, just as a reminder, they took some maps for language and also for working memory. I have all then I can show you the different regions that they took into account, but they are in blue in this figure. And they calculated the strength of connectivity, so the mean of the weight of the different tracks in these different regions, connecting these regions. And we can observe that the strength of connectivity is higher in humans when exploring the brain regions related to language. And in B, we can see that the strength of connectivity is higher in chimpanzees compared to humans when looking at the brain maps related to working memory. In particular, they explore visual working memory. And these two functions, language and working memory, were defined a priori. So they wanted to look at these different functions based on different literature that say that these two different capacities, of course, language is higher unique in the humans, and spatial working memory is better in chimpanzees compared to humans. And then in C, what they did, they wanted to explore if this network strength in the brain anatomy was more important or had a higher connectivity in humans compared to chimpanzees. So they use different maps from the Neurocino, and they explore the strength of connectivity in these different regions of interest. And we can see here in the figure 3C that all the functions that are above the dashed line are the ones that have higher network prominence in humans compared to chimpanzees. And all the terms, cognitive terms that are below this touch line represent terms that or cognitive functions that have a higher network prominence in chimpanzees compared to humans. So we can see that some of what they also discuss is that, for instance, some functions related to language and in the DMN default mode network that are more prominent in humans compared to chimpanzees, and that probably all these cognitive functions that are more prominent in the chimpanzees are related to the context of the chimpanzees, in which, for instance, for foraging, they really need all these cognitive functions to be able to survive in these more natural conditions. So these are the results, and so they also have an open question in which here if whether the chimpanzees have potentially derived this advanced network in working memory and spatial attention skill, or whether humans have the same specialization or the same strength of connectivity, but they have a larger investment in other cognitive functions that are more developed in humans, for instance, language and all cognitive functions related to the default network system. And so these are the results of this paper, showing this that there this shared brain connectivity between humans and chimpanzees, as they were able to predict cognitive scores based on the same networks of connectivity, but also some differences, because in humans we can see a higher connectivity within areas related to language, and in chimpanzees higher connectivity in areas that are more related to working memory. Some of the questions I have from this paper, and there are some limitations also that they mention, are that, for instance, most of these analyses were performed intra-hemispheric and not inter-hemispheric. And that also they were not able to predict cognitive functions related to social cognition. That's why they didn't explore this in chimpanzees, because they were not able to predict these scores within the data set in the humans. And also, this is my question, that I'm not very familiar with these comparative anatomy studies, but the fact that they use a same atlas to define regions of interest in both the humans and chimpanzees, that I'm not sure if this is something that can also impact in the results, so in the fact that they were able to predict cognitive functions from the same networks. So yeah, I just leave you discuss, or if you have questions about the paper or something that I miss, you can just let me know.
Okay, so thank you very much, Marcela. It's really interesting, this paper about similarities and the differences that we have between humans and chimpanzees. So now, fellow participants, do we have any questions? Yeah, I see.
Yeah.
Sorry.
Yeah.
Okay. I was wondering. I was looking at the correlation plot, and you can see that the effect size in humans in the correlation is like much smaller. It's like same significance, but there is way more variability in humans, and there is in the chimpanzees? Did they discuss that, about limitation, reason why there would be a difference?
No, it's true that what you say, but no, I didn't see any discussion related to the variability of the data. No. I'm just thinking that maybe it's because they calculated this composite score that involve different scores from different cognitive tasks. So I guess, yeah, it can take into account the difference in the different tests that are just an average of that. But yeah, I'm not sure because therefore the monkey is the same. But yeah, there is no discussion about this. No.
And then they use the Desikan Atlas to create connectomes and compare it between chimpanzees and humans. And because those are anatomical regions, I mean, they're not good fit with like functional territories. Do they, do they acknowledge or suggest way to do it in the future? That discussion, did you see something like that?
Yeah, no, there is no discussion related to the atlas. So I had the same question if there was some, if there is some real like analogy, but it's okay or not to use the same atlas to define both. What they discuss is only that they use the track that were present in I don't remember the percentage of the 150 out of the total set of 480 subjects in humans. But they don't discuss for the comparative analysis. There is no discussion about the atlas. And actually, I went very deep into the method because I wasn't sure if they use the same. But the description they gave is that this atlas was used to both. Okay. But yeah, no, no, they discuss more about how to compare the cognitive abilities in both to be sure that both are measuring the same, but not related to the atlas.
Right, it's still very impressive that they create like a match between the anatomy and the function in those two species.
Yeah, exactly. Yeah, it's impressive because also within humans in two different data sets, sometimes is not it's impossible to predict some cognitive functions. But yeah, in this case, impressive that they were able to do it in different species. Yeah.
Thank you, it was great.
I see another hand, Lilith.
Hi, thank you again for this paper. I mean, it was really interesting. Actually, I had also kind of the same question about figure 2, but now it's another one also connected with figure 2. So like cognitive score for humans is in one scale and for chimpanzees in another scale. So basically they, as I understand, they train the data, the model, do the prediction on one scale, and then they use that to predict another scale. I think there can be a problem here, no? Because
yeah, yeah, as I remember, they normalize the data. So
but then it's on the figure, it's like from 0.4 till 0.8. And for the humans from 60 to 140. Yeah, here.
Yeah. Yes. Yeah, that's true.
So maybe that's why if they standardize both, and then it will be a bit different, the result. What do you think?
Yeah, that's true. But yeah, actually, if yeah, you're right, the scales are not the same. So for sure, they are not so normalized. What they normalized was only at the brain level.
Yeah.
But yeah, I think it can, yeah, because if they train the model and the coefficient of correlations are prediction from brain to these scores, I don't know if it can impact actually, because they are using the coefficient of correlations. So it's like they use not the model, but values taken from a model.
But
yeah,
you're
I'm
predicting the score, right? Not the...
Yeah, exactly, the scores.
So...
But the scores are based on the coefficient of correlation between the prediction of the scores in humans.
Yeah.
Yeah, I'm not sure if it can impact. You're
Well,
right.
it would have been interesting if, I don't know, they try to do it and see if then it will be more or
maybe
less
it
the
can
same
improve.
correlation. Maybe actually humans, the correlation for humans will be like higher between predicted and actual score. No? Then
Yeah,
for
but in
chimpanzee.
humans, yeah, that's right. But in humans, the scale is the same for both data sets. So, but yeah,
yeah, I get.
Yeah, for sure. I guess it's not a problem to normalize as they are using different data sets. But yeah, I'm not sure exactly why they didn't do it. But yeah, you're right that this is something that, yeah, I didn't see. Yeah.
But actually they write in figure D normalized empirical cognitive scores.
Yeah, exactly. That's why I remember at some point I read
and
that
for the B
and
for the B they write empirical cognitive scores. Yeah,
yeah.
like in B there is not normalized, so that's what I thought. Okay. Okay, thank you.
Thanks to you.
Okay, and did they write something also about other animals?
No. No, they just mentioned in the discussion that will be interesting, of course, to explore these different network connectivity in other species. But that what is difficult is to have some comparative cognitive abilities in the different species. So this is the big issue that they mentioned in the discussion. And yeah. But and also what they mentioned, yeah, is that these skills in higher skills in the primates in working memory and spatial attention, that may be because of this adaptation in the context or to be able for the foraging. But yeah, they don't mention if they have some comparisons with other species. But it's something that they would like to do in the future.
Yeah, for sure. Yeah, I see. Okay, thank you. No more questions? All right, I think, yeah, so we conclude the Neuroccino. So thank you to everyone for attending, and yes, have an exciting science week and have a nice summer. And also, let's think, this Wednesday we have the CNS talk with Kathleen Rockland. So see ya this Wednesday. Bye-bye.
Bye.
Bye-bye.